All right, so my name is Gerhard Niederfellner, not Niederfellner. I'm founder and CEO of Beoro Therapeutics, which I will present to you with a focus on our lead asset, which is called BET-101, and it's an immunotoxin with a lot of potential for treating multiple myeloma. Beoro is a biotech startup company that focuses on developing immunotoxins, recombinant optimized immunotoxins. The founder team is very experienced in drug development, all of them working in pharma companies and biotech companies. We operate as a lean virtual biotech with only 3 FTEs and have acquired a worldwide exclusive license for our lead asset, BET-101. We also have 2 additional earlier stage product candidates, which will be for solid tumor indications. I'm not going to talk too much about them. And we're currently raising Series A financing of around 13 million euros. 90% of the proceeds will go to achieving clinical proof of concept with the lead asset. So what are immunotoxins and how do they work? Immunotoxins are recombinant fusions between an antibody fragment and a bacterial toxin fragment. In our case, that toxin fragment is a fragment of Pseudomonas exotoxin A. Our antibody fragment is a humanized Fab fragment against B-cell maturation antigen. And once it binds to B-cell maturation antigen on multiple myeloma cells, it's internalized. And then it has to undergo what is known as retrograde transport. So that brings it back to the endoplasmic reticulum. And in the ER, then the toxin moiety can escape to the cytosol, where it catalytically modifies what is called the elongation factor 2, and that irreversibly blocks protein synthesis. And then the cell either dies from apoptosis, or if it's very apoptosis resistant, it will undergo necrosis, because without protein synthesis, it cannot survive. So this mode of action will kill chemotherapy-resistant cells. It will also kill cancer stem cells, because no cell can survive without protein synthesis. And that's why immunotoxins achieve a high rate of complete responses and also very durable complete responses. I want to point out that also this mode of action is extremely potent. The therapeutic window of immunotoxins is better than for antibody drug conjugates, and that in part is due to the shorter half-life of the immunotoxins. I just want to point out here that immunotoxins are clinically validated as a drug class. So the first was approved in September 2018. That is a molecule called Lumosity. It targets CD22, was developed by AstraZeneca, and it's being used to treat chemorefractory hairy cell leukemia patients. In the pivotal phase 2 trial, 63% of the patients could receive 6 cycles of treatment, and that resulted in an objective response rate of 75%. But more important is that 41% of all patients had a complete response, and 30% even had a durable complete response, which in the study was defined as no tumor cells detectable in the blood or bone marrow for greater than 6 months. And we are convinced that multiple myeloma is the next highly promising indication for such a PE-based immunotoxin. So what is multiple myeloma and where is the medical need? So multiple myeloma in principle is a treatable disease, but the problem is that everybody relapses. And with every relapse, the response to current treatments gets shorter and shorter. So if a patient, for instance, is resistant to proteasome inhibitor and an immune modulatory drug, that patient typically survives only for 8 months. And so the primary goal in multiple myeloma therapy is not to have a high rate of responses that don't last very long, but the goal has to be to get a high rate of complete responses and very durable complete responses. And that, we think, is where the immunotoxin comes into play. In terms of market opportunity, so multiple myeloma is the second largest hematological cancer indication. It affected in 2016 about 280,000 patients in the 7 major markets. If you include China, that number goes up to 800,000 because the Chinese population is old, and multiple myeloma is a disease of the elderly. Despite those numbers, it's designated an orphan disease, so you get market exclusivity after approval. And of course, why it is interesting also is it's a big commercial opportunity, and that's probably best illustrated by the example of daratumumab. It was FDA approved in 2016 as second-line treatment. In 2017 already, the net year sales, full year sales, totaled $1.2 billion. And I want to point out that in the future, the costs for new drugs will be partially offset because major agents go off patent. So Revlimid goes off patent in 2026, Velcade goes off patent in 2022. And Revlimid in 2017 was the most expensive Medicare Part D drug with over $3 billion. So there is money going to be released. This is what our optimized immunotoxin format looks like. So we use a humanized Fab fragment that targets BCMA. BCMA is the very highly validated selective target for multiple myeloma. It's a nice target for a payload approach because the multiple myeloma cells depend on its signaling for survival and proliferation, so they cannot easily downregulate it. And we have found that it's a very optimal cargo carrier for an immunotoxin because it allows efficient escape to the cytosol because it shuttles back between the cell surface and the ER all the time. On the payload side, we use only the domain 3 of the exotoxin, and by eliminating the domain 2, we get rid of a lot of T-cell epitopes, so the major T-cell epitopes are in domain 2. We get rid of a lot of protease sites, and for reasons that are molecularly not understood, we have a much better tolerated molecule. So the class effect of immunotoxins as an off-target effect is vascular leak syndrome, which has been dose-limiting also for Lumosity. When we tested a molecule without domain 2 in a rat model of vascular leak syndrome, we could dose it 10-fold higher without getting vascular leak syndrome. Also, we know from the preclinical data in the mice where you only see off-target effect, but in the mouse, our molecule is tolerated at 8-fold higher doses than Lumosity, for instance. Our IP, as I said, rests on the binders. So we have licensed the IP to the CDRs of 2 anti-BCMA binders from the NIH. This binder IP expires in the US in 2035 and in Europe in 2038. We also licensed the IP on using this truncated toxin fragment, but that IP expires a little earlier in 2030. And our license allows us not only to develop the molecule for multiple myeloma, but for any blood cancer that has high levels of BCMR. So that would include, for instance, diffuse large B-cell lymphoma, acute myeloid leukemia, or classical Hodgkin's lymphoma. At the end, I just want to show you very good in vitro and in vivo efficacy data that we received with this molecule. So in vitro, when you test it on established multiple myeloma cell lines, but also on primary patient cells, which is more relevant for the clinical translation, the average IC50 is 80 picomolar. What is important is that when you only incubate the cells for 10 minutes with the immunotoxin, 89% of the cells die. So you don't need a long exposure. And that speaks to the efficiency of BCMA as a cargo carrier for the immunotoxin. And we had the chance to test the molecule on patient cells from a patient at first diagnosis and after he relapsed on the combination of proteasome inhibitor and immunomodulatory drug, and the IC50 did not change. So the cells did not become resistant, which to us indicates when we go into second line, we will get a good response rate. The molecule has been tested in 2 disseminated xenograft models of multiple myeloma. The data of one of the models is shown here. It's the H292 model. And the mice received 5 doses given every other day. And as you can see at the bottom line, so basically all mice were tumor-free from day 11 onwards until the end of the study at 3 months. The vehicle-treated control mice developed increasing tumor load, and typically those mice have to be terminated at day 40 or 42. The other model, MM1S, is even more impressive because we and others have shown that that model is resistant to treatment with lenalidomide, bortezomib, or dexamethasone, so to all the standard of care treatments in multiple myeloma, again pointing to the fact that we will have good response in second-line treatment. So then, of course, the big thing that all the VCs always point out is it's a very competitive field. That is true. We looked at the competition. About half of the competition is still preclinical, but there is some very advanced approaches. The competition is mostly on T-cell-based approaches. So 80% of all the approaches are either a CAR T-cell approach or a T-cell recruiter approach. And the problem I personally have with these approaches is A, they have a difficult to manage safety profile, and B, they cannot be combined with any of the standard of care treatment agents. So unless they on their own cure the disease, they will not be good. For CAR T-cell therapies, we know already that there's no cures in multiple myeloma with CAR T-cell therapies because even the best responding patients relapse after 1 year. If you take that together with the cost, the price tag of a CAR T-cell therapy, and the logistic difficulty, I think it's a no-go. For the T-cell recruiters, they have a similar objective response rate, but one has to point out that they only get robust clinical responses at the maximally tolerated dose, and at that dose, 40% of the patients have to be hospitalized for serious adverse effects. And Paul Richardson, the head of the Multiple Myeloma Center at the Dana-Farber at the recent ASCO, pointed out that managing clinical safety for these molecules is going to be a real issue. And finally, the other competitors are the ADCs, and many of those use payloads that are either clinically unproven, like alpha-amanitin, for instance, or that have failed already several times in the clinic, like the pyrrolobenzodiazepines that AstraZeneca uses. So we think that the most relevant competitor is the GSK molecule. It has a relatively good objective response rate of 60%. It will likely be approved next year by the FDA, but the weak point of that molecule is has only a 16% complete response rate, and we know that the duration of response is only 14 months, which is good for an ADC because all the other clinically approved ADCs have only 7 months. This one has 14 months. But if you compare it to Lumosity, that has greater than 30 months clinical complete response duration and has a 41% complete response rate. And I think those two parameters are important differentiators for multiple myeloma. So I want to thank you for your attention, and I'll take any questions that you might have.